This unit covers pathogenic microbes. Most containment facilities deal with microbes in various ways for various reasons. Since you'll not be working directly in these areas, knowledge of them is for your safety and peace of mind. Move through these slides at your own pace. Some of them will be straightforward, but if you see the microphone image on the slide, I'll be making a few brief comments to explain terms and concepts. Looking at these learning goals could be intimidating, but if you believe that knowledge is power, well, this power is going to give you peace of mind and help you stay safe. That's all. Let's go through these terms quickly because there are written definitions on the next slide. If an organism is pathogenic, that means it causes a disease. An example of this would be Streptococcus pyogenes. That's the bacteria that causes the really bad strep throat we get. If an organism is not pathogenic or non-pathogenic, it just means it doesn't cause a disease. There are hundreds of thousands, probably more than that, bacteria that do not cause disease. Lactobacillus is an example of a non-pathogenic bacteria. That's the bacteria we find in our yogurt. There are bacteria that have strains that are pathogenic, E. coli, the E. coli that we hear about when food is recalled. But E. coli is also found in our gut. It's a good bacteria there. It's non-pathogenic. Virulent, it means how easily an organism can cause a disease, and that disease is going to be a bad disease. It's going to cause damage. So when we talk about very virulent organisms, we might be talking about Ebola or TB. These are pretty easy to get, and they cause damage to the host. A hot area or a hot lab usually refers to an area that has radioactive materials in it, and you don't want to go in without the right kind of protection. But sometimes those terms are used to describe labs that have pathogens. Those labs are also referred to as live areas. Mode of transmission is the term used to describe how a pathogen moves from the area where it's growing by a specific route to a new host, and that's you. Sometimes this is called chain of infection. We hear so much these days about disease and epidemics that it just seems fair to talk about the benefits of microorganisms. So I'm just going to give a few good examples. Microorganisms are important for good health. We need good body flora, a lot of good body flora. And microorganisms are important in medicine. Antibiotics, insulin, growth hormones are all mass-produced with the aid of microorganisms. They're used in food production. Yogurt, bread, cheese, beer, all of those depend upon microorganisms, bacteria, and funguses. Microorganisms are used in chemical production of enzymes and color pigments, biofertilizers, and biofuels. They're used in genetic engineering to correct gene defects. And again, the production of insulin is dependent upon microorganisms. And then a really good thing is that they are used in environmental engineering. There are bacteria that are used for the degradation of toxic chemicals, like as in oil spills or plastic or water waste. Let's talk for just a second about different types of microorganisms. We'll start with bacteria. They can't be seen with the naked eye. You have to have a microscope to see them. They're unicellular, one-celled, but they have some type of cell structure. That's good because that means antibiotics, antiseptics, disinfectants can kill them. Viruses are smaller, much, much smaller. They can only be seen with an electron microscope. There really is no antibiotic that will kill them. Really isn't. You can kill many of them with an antiseptic or a disinfectant, and certainly with heat. Fungus or fungi are organisms that reproduce using spores. So what that means is that they can be molds or yeast, easily killed again. Parasites are microorganisms that have to have a host to live. So they can't live by themselves. They live off of something else. Some of them are really quite difficult to kill. And then this is a microorganism that is not mentioned on the slide, but you'll hear about it. Prions. Prions are very small. There's a lot of debate over if they're alive or not because they don't have structure, they don't have a nucleus, they don't have DNA or RNA, but they are proteins and they cause a reproduction. They are the cause of mad cow disease, and that's very bad. Emerging diseases are the diseases probably being researched and worked on in the buildings you're working in. These are not new diseases, not caused by new bacteria or viruses. It's just that their causative microorganisms have undergone distinct changes, making the new disease something we've never seen before. Zika virus is an example of that. The virus has probably always been around, but has undergone a lot of change, causing a disease that we've never seen, never needed to study, never needed to know how to combat. So Zika is a disease that we would say has recently arisen. There are diseases, though, that are called emerging diseases because they've spread beyond their local or their original location. Ebola would be an example of that. In its original location, it was spread by monkeys, and the spread currently is being caused by humans. So it is an emerging disease that's moving across miles and borders. There are some diseases called emerging diseases because they've grown more virulent. An example of that would be TB. TB has become not susceptible to antibiotics. So to combat it, we call it an emerging disease and look at the eradication of it in a different way. Select agents are the microorganisms and toxins that pose the greatest threat to health of animals or humans. They're very highly regulated by a group out of the CDC, the Federal Select Agent Program. That program maintains a database where all of these microorganisms are, how much is in those locations, who's using them and for what purpose. The program inspects the facilities that use these select agents or that transfer them. They ensure that all people who are working with the select agents undergo a very strict security risk assessment. So you can see there's quite a bit of regulatory oversight, quite a bit of paperwork that is involved with the use of these select agents. Examples of them, though, might give you a hint as to really how dangerous they are. The bubonic plague is a select agent. Smallpox is a select agent. And then an example of a toxin would be ricin. So, should you be concerned about working around microorganisms? Maybe, maybe not. I think the real answer has to do with how informed you are about them and how you apply that information so that you can work safely. The next few slides are going to shed quite a bit of light on working safely with or around pathogenic microorganisms. It's not the non-pathogenic that are going to cause us problems, but microorganisms that are pathogenic are what are going to make us sick. So there are some criteria for the successful pathogenic microorganism. It has to get to you. In other words, the term used is there has to be a successful mode of transmission. It has to infect you, so it has to get into your system or contaminate your skin. It has to overcome your immune system so that you become sick. One cell, or probably even a colony of pathogenic microorganisms, are not going to make you sick. It has to be able to divide and grow to cause illness. And then it has to spread, and honestly, it has to be able to move to a new host to be considered a successful pathogenic microorganism. The next slide is a graphic showing what we call the chain of infection. Starts with the reservoir and moves all the way through the infected host. Chain of infection is just another way to describe how pathogens work. If we follow this diagram from upper left down to the lower right, and we start with infectious agent, that's just the pathogen. An infectious agent is the pathogenic microorganism, but it's got to have an environment it can live in. Water, soil, like with anthrax. Sometimes the first person who has the microorganism, say the person you caught the flu from, would be the reservoir. And then pests are sometimes reservoirs. They're good environments for the microorganism to grow, but they transmit those microorganisms to us. The mode of transmission is how they do that. Sometimes it's an aerosol, like when you cough or sneeze. Sometimes it's contact, when you touch a solid surface and you literally come into direct contact with the bacteria or virus, sometimes even a parasite. There has to be, though, an entry portal. So you've directly touched a surface and you rub your eye, or you eat something that is contaminated, or it's just on your skin and it's absorbed. That's called the entry portal. And then when we move on down to the very last image, the susceptible host, well, some people are just more susceptible. They're more susceptible to disease. Their immune system doesn't handle infection the same. So all of this goes together to say, if we're safe and we practice good safe technique and we're vigilant, there should be no reason why we would become infected. We covered the different levels of biosafety labs in the first unit. This slide is just to remind you of those levels, beginning with BSL 1. That's just about like your kitchen. Nothing pathogenic is used in there, all the way to BSL 4, where the most virulent pathogens are worked on. It probably doesn't need to be said, but the absolute two best methods of prevention are to regularly and frequently wash your hands and to remain vigilant to apply what you're learning here. It may seem unnecessary or inconvenient, but complying with your facility's medical surveillance plan is very important. You likely had a physical when you began your employment. That is what provided the baseline for several indications of health. Minimally, you should report fevers, chills, dizzinesses experienced while at work. Beyond that, your HR or your biosafety officer will acquaint you with the details of the facility's medical surveillance plan. Every facility that is working on or with microorganisms is required to have a biosafety officer. This is the person who writes the policies for safety and handling microorganisms. This is the person you would go to with your questions about your own health or monitoring your health, exposure issues. This is always the person to alert first in case of an emergency.